Hi friends, today we will be talking about one of the pathways operating in C3 plants, especially under the conditions of higher temperatures, and that is what is called as photorespiration. More popularly called as photorespiration, but has nothing to do with the cellular respiration actually, because whenever we talk about respiration, we think about conservation of energy and production of energy in the form of ATP. This particular pathway, the photorespiration, has nothing to do with conservation of energy in the form of ATP. Rather, it results in the loss of energy. It results in the loss of the carbon dioxide, which is being fixed with a lot of efforts in the Calvin cycle. So talking about this particular pathway, so it is known by other names also. We refer to this pathway as the C2 pathway, or oxidative photosynthetic carbon cycle is one of the alternative names for it, or we also refer to as the glycolate pathway. Now, as per the norms of the biochemistry for naming the pathways, we generally label it according to the first product which is formed. So here also you have the first product that is formed is phosphoglycolate converted to glycolate, and therefore the pathway called as the glycolate pathway. This particular glycolate formed is made of 2 carbon atoms, so therefore this is called as a C2 pathway, and it results in oxidation. So oxidative photosynthetic carbon cycle. Actually what happens here is, this is a pathway where we are actually trying to convert this metabolically useless product, that is phosphoglycolate or glycolated 2 carbon intermediate, which is of no use to the cell, to the formation of serine. So we have these 2 molecules of 2 carbon each, converted to your serine, which is actually a 3 carbon compound, and during the process there is liberation of carbon dioxide. So oxygen enters in and carbon dioxide leaves out. So therefore probably to have a simile there, we call this particular mechanism as the photorespiration, because we generally refer oxygen coming in and carbon dioxide going out as the respiration, and this takes place in the presence of light, so we call it as a photorespiration. But then it may lead to misnomer, like it may lead to some wrong concepts, and therefore the alternative names for it, the glycolytic pathway or the oxidative photosynthetic carbon cycle. Where does it all start? It all starts when your Rubisco, you remember this enzyme Rubisco, ribulose 1,5 bisphosphate carboxylase oxygenase. Not heard of much, right? So not heard of much means we never elaborate this name. We always refer to as the Rubisco. Rubisco basically has the carboxylase activity, which is much talked about, because that is the feeding enzyme, you can say, which is fixing the atmospheric carbon dioxide into the organic world and feeding all of us. So one of the enzymes which is working most abundant enzyme in the biosphere, which is feeding all of us on this particular planet, it is your Rubisco. Its alternative activity, that is of oxygenase, this is what is in picture in this particular pathway. So ribulose 1,5 bisphosphate carboxylase oxygenase, when it shows its oxygenase activity, where instead of combining with carbon dioxide, it combines with your oxygen. So if you recall the Calvin cycle, which we have studied in our earlier lectures, where ribulose 1,5 bisphosphate combines with carbon dioxide. So 5 carbon ribulose 1,5 bisphosphate, 1 carbon dioxide, you have 6 carbons, and 2 molecules of 3 phosphoglycerate are formed, each of 3 carbon atoms. This is what happens actually. But then if it combines with oxygen, you have a net of 5 carbon atoms only. So you have 1 molecule of 3 phosphoglycerate which will enter into the Calvin cycle, it will operate smoothly, but one particular product which is formed is your 2 phosphoglycolate, which is of no use as such to the cell. Now we need to salvage this back, because if we don't salvage this back, we lose everything what we have fixed. So this 2 phosphoglycolate is dephosphorylated to form what is called as your glycolate. Now this glycolate, we know that this is operating in the chloroplast stroma. One of the another characteristic feature of this photorespiration, since it is the most popular name, I will be referring to that. So this particular pathway operates in these 3 compartments, cellular, what you call as organelles. So one particular aspect, when Rubisco, instead of combining with CO2, if it combines with oxygen, which generally happens at warmer climate, at higher temperatures, when the partial pressures of carbon dioxide are a bit less and oxygen are more. So there all these 3 organelles, they are put into work. So your glycolate, which is formed in the chloroplast stroma, that is transported into your peroxisome. Now where it is oxidized by the glycolic acid oxidase, with intermittent formation of H2O2 to form glyoxylate. So your glycolate is oxidized to form what is called as your glyoxylate, a 2-carbon intermediate, which is then transaminated from the serine here to form glycine. Now this set of reactions occurs in the peroxisome. Glycine formed in the peroxisome is now transported to your mitochondria. So in the mitochondria, these 2 molecules of glycine, so you have one pathway, then the similar process occurs, again another molecule, so you have 2 molecules of glycine. So these 2 molecules of glycine now, they are acted upon by glycine decarboxylase. So it catalyzes its what you call as oxidation and what you call as removal of carbon dioxide, removal of ammonia to form what is called as your serine, a 3-carbon compound. So 1 carbon is lost here and you have 2 units of 2 carbons. So otherwise you would have lost all the 4, you have got back 3. So you can say that you have saved around कितना 4 में से 1 loss हुआ, 3 save किए, so you have saved around 75% of your carbon, you have lost only 25% here. So serine, when we are talking about this serine, once it is formed in the mitochondria, this is transported back from where it had come. They talk about it, glycine कहाँ से आया था, तो they say it came from peroxisome, so serine is pushed back to peroxisome. This again by transamination is converted to hydroxypyruvate acted upon by alpha hydroxy acid reductase to form glycerate. So there is a reduction of this particular hydroxypyruvate to form what is called as a glycerate. Glycerate now pushed back to from where it had come at the very first place, that is your chloroplast. So your glycerate, again a 3-carbon intermediate, brought back into your chloroplast stroma, where this is phosphorylated to form what is called as your 3-phosphoglycerate, which can again enter the Calvin cycle. So हुआ क्यों ये सब, मतलब why this happened at the first place? To get back these 3 carbons. This pathway, if it had not operated, we would have lost around 4 fixed carbons. But because of this particular pathway, we salvaged back. Out of these 4, we brought back 3. So you can say that 75% of your carbon you have brought back, you have read to the 25%, but then still this is very costly because it requires the expenditure of ATP, and therefore organisms which are living, like plants which are living in such warm conditions, they have adapted to, resorted to different pathways like your C4 pathway, some have resorted to the CAM pathway, because combining of your ribulose 1,5 bisphosphate carboxylase oxygenase, the Rubisco, with oxygen, बहुत महंगा पड़ जाता for this particular cell. So therefore we have evolved, not we, like we don't do anything, we are consumers. So this is a part of this photosynthesis. So some of the plants, like we refer to as C4 plants and CAM plants, they have overcome this problem of photorespiration. So that we will see in our upcoming lecture. So here you can see in photorespiration what has happened. Your Rubisco, instead of combining with CO2, it combines with oxygen. Very clear. So photorespiration, like uska onset kab hota hai? When Rubisco, instead of combining CO2, combines with oxygen. You have 1 3-phosphoglycerate, 1 phosphoglycolate. Phosphoglycolate converted to glycolate. Glycolate transported to peroxisome, oxidized to glyoxylate, converted to glycine. Glycine transported to mitochondria. 2 molecules of glycine converted to serine. 1 CO2 is released, serine transported back to peroxisome, converted to hydroxypyruvate, then reduced to glycerate. Glycerate transported to your chloroplast stroma, and there phosphorylated to form 3-phosphoglycerate to again operate this particular Calvin cycle. Stay tuned with me, Professor Girish Kukreja, for more on C4, CAM and more in photosynthesis. Thank you.